So let's do meiosis. Remember we had done mitosis.
Yeah.
Yes, we had done mitosis, right? So mitosis was equational division, right? That means the cells, whatever the chromosome numbers, they were equally divided. What we are studying here is meiosis. Okay, so the term meiosis was given by Farmer and Murray and Farmer. Murray and Farmer in the year 1905. Meiosis is a type of cell division that takes place in the gonads. In the gonads, that is the sex organs. We know that the sex organs are involved in the formation of gametes. Okay, like for example, testis. It is a gonad. It's a sex organ, it's a gonad, and it is involved in the production of sperm. Ovary. Again, a gonad and a sex organ involved in the production of ova. This is the male gamete. This is the female gamete. Okay. Now, here, the gametes, you have to remember one thing. The gametes, the chromosome number of gametes, is always half the chromosome number of the other cells. That means our other cells has 46 chromosomes. So on the contrary, the gonads have half the number of chromosomes, that is 23. 46 means 23 pairs. So this is our gametes. Now, why, Sana, can you tell me why do gametes have half the number of chromosomes?
Because
yes,
it's
because yes, see, male parent, female parent. Okay, male will produce male parent, female parent chromosome number is 46. Male will produce sperm. Female will produce ova. Female will produce ova. So, now, suppose if the sperm is 46 chromosome number, ova is 46. These will fuse in the process of fertilization to form zygote. Okay, they fuse to the fertilization to form what? Zygote. Now, if the zygote, that means if sperm has 46, ova has 46, this will combine. So, what would be the chromosome number? Can you tell me? 46 plus 46, how much?
That is a 92.
Yes or no?
Yeah.
Yes. Is it possible? Is it possible that we have 92 chromosome number?
No.
No. So that's the reason that when the gametes are formed, the chromosome number is reduced. If the individual has 46, 23 pairs, then gametes will always have only 23 chromosome. Here also 23 chromosome. So these individuals, these are called as diploid. Diploid. whereas the gametes will always be haploid, n number of chromosomes. Only when we have 23, 23, and in the zygote we can have 46. So now the chromosome number is maintained. So we will have all throughout a constant chromosome number.
Okay.
Point is clear?
Yeah.
Okay. Next, let us see. Okay. Now, meiosis only occurs in the gonads. Meiosis doesn't occur in any part of the body. Whatever cells are there in other parts of the body, okay, other parts of the body, that will undergo mitosis. Okay, so these will all be meiosis. So meiosis reductional division. That means here the chromosome number is reduced. Chromosome number is reduced. Okay. Now, what was mitosis? Mitosis is equational division. Mitosis is what division? Equational division. And meiosis is what? Reductional division. Chromosome number is reduced. When we talk about meiosis, meiosis has two divisions. Meiosis 1 and meiosis 2. Meiosis 1 will have the stages. Okay. Meiosis 1 will have prophase 1. Okay, meiosis 1 will have prophase 1. After prophase 1, we'll have metaphase 1. Then we'll have anaphase 1, telophase 1. Okay, telophase 1. Between telophase 1 and meiosis 2, there is a resting stage called as interkinases. Interkinases is resting stage. Here, there is no DNA replication. Here, there is no change. No change over chromosome number. Okay, is this clear?
Yeah.
Oh, all right. Okay. Okay, let's see this. Okay, no change in chromosome number. Now comes meiosis 2. Meiosis 2 will have same prophase 1. Okay, can you tell me the other stages?
Metaphase.
Yes, metaphase. Metaphase 1 or 2? This is 2, sorry.
Yeah, 2.
Mm-hmm.
Metaphase 2.
metaphase 2. Then
anaphase 2.
anaphase 2. Then
Telophase 2.
yes, telophase 2. Okay. All right. Now, see here. Meiosis 1. This is the one where the actual reduction of chromosome number takes place. Okay, meiosis 1 is the actual reductional division. Prophase 1 is divided into 5 stages. Leptotene, zygotene, pectine, diplotene, and diakinesis. Have you done this?
No, not yet.
Not yet. Okay, what have you done in meiosis? Just the starting, right?
Yeah, and also like the what happens in each stage.
Hmm.
Oh, and what's it called? Oh, independent.
Assortment.
Yeah.
Okay, no problem. Here we have, the book is there with me. So we have prophase 1. We will do it, no problem. So I'll just do this in detail, okay?
Yeah.
Yeah.
Oh, and also like non-disjunction.
Non-disjunction, okay. That comes later. First, let us finish the meiosis, okay?
Mhm.
Okay. Prophase 1, metaphase 1, anaphase 1, and telophase 1. It is in anaphase 1 that the actual reduction takes place. 2n to n. Okay. At the end of telophase 1, we will get, sorry, at the end of telophase 1, we will get 2 cells. And both these cells are haploid, having half the number of chromosomes. Meiosis 2 is similar to mitosis. In meiosis 2, we'll not say it's an equational division, but it maintains chromosome number. That means it does not allow the chromosome number to go below 23. Okay. So at the end of telophase 2, we get 4 cells. Okay. At the end of telophase 2, we'll get 4 cells, each of which is a haploid. Okay, each of this which is a haploid. Okay, so let us do one thing. Let us let us see the process of meiosis. Okay, so let me see here. Okay, fine. Now, so when we talk about the meiosis, okay, when we say meiosis and all the different chromosome number, how it takes place and all that, okay, so let us see the different stages of meiosis. Okay, different stages of meiosis, and we clearly study all the stages of meiosis. Okay, so here I think in your textbook you have the book that you have, the reader, page number 164 and 65 has the diagram of meiosis. Okay.
Mhm.
All right. So let's see. Okay. So in prophase 1, let us see meiosis 1. So the first stage is prophase 1. So in prophase 1, if we have, this is the nucleus. The nucleus is retained. This is the centriole. The nucleus will be retained. Okay, prophase 1, the first stage, which we call as leptotene. In leptotene, this is one chromosome. This is the other chromosome. So in leptotene, you'll see the arrangement of the chromosomes. That means here, the chromosomes become condensed, and you can easily see the chromosome. In that, when we talk about in the leptotene, the next stage is the zygotene. In short, I will explain you. Zygotene. Suppose, for example, the red chromosome comes from the mother, the blue comes from the father. A nuclear membrane is still there. So we will have the chromosomes in zygotene that will be placed together. And they are placed together by a strand here, which has a lot of RNA. This is called as the synaptonemal complex. This complex will keep both the chromosomes together. In the pectine, we'll observe a very important feature which is called as crossing over. Crossing over means exchange of genes. You can see here. See this. Okay. So in pectine, we will see crossing over. Okay. And this is nothing but exchange of genetic material. We say no variation. That means those who reproduce sexually. We have some features of our parents, but we are not exactly like our parents. That is because of crossing over and exchange of genes. This crossing over will bring about variation. Okay, will bring about variation. Clear?
Yeah.
Okay, this will bring about variation. Now, next, let's check on this. Pectine. Okay, so now the crossing over is between two non-sister chromatids or different two chromosomes. After pectine, the leptotene, zygotene, pectine, we have diplotene, and at last we have diakinesis. So by the time the cell reaches in diplotene, or by the chromosome reaches in diplotene, the crossing over will be completed. Diplotene and diakinesis, both of them. The crossing over will be completed. So you can see that the colors have exchanged, indicating that the crossing over has completed. Okay. And once the crossing over is completed, these chromosomes will separate in diakinesis. Once they separate, now the cells at the end of that, after dikinesis, what happens? After dikinesis, we have the next stage, that is metaphase 1.
Mhm.
Metaphase 1. In dikinesis, nuclear membrane and nucleolus disappears. Nucleolus disappears. Okay, nuclear membrane, nucleolus disappears. Now, next, metaphase 1. So, Sana, can you tell me what happens in metaphase 1?
It's the cell, like,
What
the
happens in
pairs,
metaphase 1?
the pairs go to the different poles of the cell.
No, they don't go to the different poles. Yes, can you tell me what happens? Yes.
Is it the...
See here. There's one chromosome. There's another chromosome. Remember, it was exchanged.
Yeah.
Now, centrioles are present here. So, centrioles will send out. This is the centriole. This is the spindle fibers. Okay. So now what happens? These two chromosome, okay, Sana, this two chromosome, one is from the mother, one is from the father, right? This two is called as homologous chromosome. So in metaphase 1, there will be two metaphasic plate, one chromosome lying here and below that another chromosome. This is the condition metaphase 1. Then we come to anaphase. Anaphase 1. In anaphase 1, the chromosomes are pulled apart like this. Okay, chromosomes are pulled apart. And who pulls them? The spindle fibers. Okay, so chromosomes are pulled apart. Okay.
Yeah.
Once they go to anaphase 1, after that we have telophase 1. Okay, so telophase 1 will bring the chromosomes to the opposite poles. Okay, we'll bring the chromosomes to the opposite poles. Do you understand, Sana?
Yeah.
Okay. Here in telophase, here in anaphase 1, telophase 1, what happens here? Chromosomes will undergo decondensation. That means, decondensation means these will become thread-like. Okay, these will become thread-like. Okay, thread-like again. And these are nothing but they are called as chromatin fibers. Okay, chromatin fibers. Okay, next. Around this chromosome, gradually nuclear membrane and nucleolus will reappear. Nuclear membrane and nucleolus reappears. As a result, what we will see is that now at the end of telophase 1, there are 2 cells. Okay? There are 2 cells. Okay, so the 2 cells with the chromosome. Okay, with the chromosome number haploid. This will form this, another will form this. So here you can see that at the end of meiosis 1, we are getting how many cells? We are getting 2 cells. On the similar note, same thing, similar, no difference. We will see we'll see the division that will control the chromosome number or will regulate the chromosome number. So let us see here. We will see, 1 second. 1 minute. Okay. We will see meiosis 2. In meiosis 2 now, each cell that was formed in meiosis 1, that is going to go forward and form 2 cells. So, we shall have prophase 1. Okay, we shall have prophase 1. So, this would be, sorry, we shall have prophase 2. Chromosomes and will consist of chromatids and they are in the condensed state. They are in what state? Condensed state. After that, the nuclear membrane and nucleolus will gradually disappear. Finally, this is prophase 2. Then we have metaphase 2. Okay? In metaphase 2, all the chromosomes will align in the equatorial plate, like this. Okay, all chromosomes will align the equatorial plate. And then here, after aligning into the equatorial plate, you see, in anaphase 2, we will only have the chromatids being separated out like this. Chromatids are separated. Okay, chromatids are separated. In anaphase 1, homologous chromosome separated. In anaphase 2, okay, chromatids, that is arms of chromosome, separated. Please read these 2 lines, Sana, and tell me, have you understood? Is there anything which you have not understood? Is there a doubt or anything? See, have you understood the difference between the anaphase of mitosis?
Yeah.
This is what we said. This is anaphase of mitosis. This is anaphase of
Meiosis.
okay. See, any doubt here?
No.
Yes, see, anaphase of a homologous chromosome separate. This will separate. And anaphase 2, chromatids arms of the chromosome separate. Okay. So after anaphase, we'll have the telophase. And in the telophase, we will have 4 cells. Okay. At the end of meiosis 1, we had 2 cells. At the end of meiosis 2, we'll have how many cells? 4 cells. This point is clear?
Yeah.
Yes or no? Yes?
Yeah.
Yeah. Okay. What we are dividing in meiosis is chromosome. Keep that in mind. Where are chromosomes present, Sana? Can you tell me?
In the DNA, in the nucleus.
Sorry?
In the DNA.
Yes. Where are chromosomes? Yes. They're present in the nucleolus. Yes or no?
Yeah.
Sorry, nucleus.
Yeah.
Chromosomes are present in nucleus. That means in the nucleus, the chromosome is present in the form of chromatin fibers. If we look into the cell, this is the nucleus. Here we will have chromatin fibers. What we'll have? We'll have chromatin fibers. Now, this chromatin fibers, they will undergo condensation. That means they will become thick and more prominent. You can easily see the structure. Condense to finally form chromosomes. If we look into a simple structure of chromosomes, this is what the chromosomes are. This is called as the chromatid. Chromatid means arms of chromosome. Chromatid means arms of chromosome. Okay. This is primary constriction, or it's called as the centromere. Okay. This is the arm of the chromosome. And then we have the centromere. These points are clear, Sana? Any doubt here?
Yeah.
Okay. Inside this chromosome is a highly coiled DNA. DNA is called the genetic material. DNA is called as what? Genetic material. It is the DNA that will store all the information that forms the entire organism. What is our eye color, our skin color, our characters, everything is determined by this DNA, which is present in a highly coiled manner inside the chromosome. Chromosome has a thin membrane which is called as pellicle. This is membrane of chromosome. Inside that, there will be fluid. And in that fluid, we have the DNA. The other name of DNA is also called as chromosome. Okay. So this is how the DNA, this is a place where the DNA is present. Always remember, our DNA is always associated with histone proteins. It is the histone proteins that is always present in DNA. We will never find a DNA without a histone protein. Okay. Now, DNA features we have studied. Okay. All right. So now, if I am saying that DNA is the genetic material, DNA stores information. So what will be there in the in the DNA is base pairs. We had done, right, this one, base pairs. And it is this nitrogen base pair that is storing the actual information in DNA. Keep this in mind. Base pairs, or I will say nitrogen. Nitrogen base pair is actually storing information. Okay, storing instead just genetic information. Sana, this point is clear or not clear?
Yeah.
Yes? Okay. Now, so now when we talk about gene, it is a segment of DNA that stores information, or rather, I will write genetic information. Store genetic information. This point is clear?
Yeah.
Okay. So now, our DNA is present inside. So see, every gene is nothing but having a nitrogen base pair. Complementary base pair will be there. Okay, and these base pair will help us to recognize the different genes. Now, see here. For example, let's see. Okay. Let's see here. This is a chromosome. Okay. This is a chromosome. And our DNA is present here. So, suppose our gene for height, or sorry, skin color. We have a specific gene for skin color. That is the reason why we are having the skin color. Whatever information is there in our DNA, it is expressing itself in us. Okay. Whatever information is there in the DNA, in the nitrogen base pair, it is expressing. So now, if it is not expressing, then we can't have a skin color. We can't have an eye color, our curly hair or straight hair. All this will not be there if our gene is not expressing. So Sana, you have done replication, you have done transcription. Can you tell me what are the methods through which gene can express itself?
What are the methods?
Sorry?
Sorry, can you repeat?
What are the methods through which gene expresses itself? See, without expression, there's no character. Yes. You have done transcription. Yes.
Yeah.
RNA synthesis. You have done translation, protein
Yeah.
synthesis.
So it's
Yes. Transcription,
transcription.
protein synthesis are different ways through which gene expresses itself. Did you get it?
Yeah.
Okay. So we'll see. Transcription. Translation. These are two methods by which gene express. Okay, gene express. Now suppose, for example, gene for skin color is present here. Okay. Now, this is the position. This is the position where the gene for the skin color is present in every one of us. Skin color, gene of skin color cannot be present here, cannot be present here. It is present only here. So it's like, it's like you stay somewhere and your postal address is there, your address is there, right? In the same way, where the position where the gene is present, that is like the address or the place where the gene is found. If I am looking into a gene for skin color, I should look into that place only. I cannot look anywhere else because gene for skin color has this specific position. Okay, gene for skin color has this specific position. And this position of gene, okay, is called as locus. This is the position of gene on chromosome. Now you see, we have two arms of chromosome. I have drawn the red line in both the arms. Can you see that?
Yeah.
Yes. Why did I draw in both the arms? Can you tell me?
Because.
Yes. Think. Think. Why did I draw this line on both the arms of the chromosome? That means, what does it mean? The gene has its another pair, yes or no?
Yeah.
Yes. The gene occurs in the form of pairs. One pair will be in one arm of the chromosome, another will be in the other arm of the chromosome. This alternate form of gene, alternate form of gene is called as allele. What is it called? Allele. Alleles are alternate form of gene. Alleles can be homozygous. Okay, alleles can be homozygous. Now, when Mendel did his experiment, he said usually, we have two different characters. One will be dominant character. Another one is recessive character. Dominant means this gene will not allow any other gene to show its expression. And recessive is, it's a weaker gene that gets suppressed, that will not show its effect. So allele can be homozygous if dominant, suppose T, capital letter T, capital letter T. So both are capital letters. When it is capital letter, it means it is dominant. Homozygous, keep in mind. Homozygous, recessive, small letter T, small letter T. Alleles can be heterozygous. Alleles can be what? Heterozygous. Okay. Heterozygous means one capital letter and one small letter in the same chromosome. One capital and one small. So suppose, for example, capital letter T stands for tall plants. Small letter T stands for dwarf plants. Now you tell me one thing, Sana, here. Heterozygous, one capital letter, one small letter. What would be the phenotype or what would be the character of the zygote?
Tall plant?
TT, tall plant. Yes?
Yeah.
Why? Because one capital letter T is there.
Yeah.
And this capital letter T is dominant.
Mm-hmm.
Okay, this is dominant. And the small letter is recessive. So since we have one dominant gene, the character will be tall. This point is clear?
Yeah.
Okay, any doubt here?
No.
Okay. So, alternate forms of genes. So, allele can either be homozygous, that means homozygous dominant, or homozygous recessive. Allele can be heterozygous in this form. Okay. This did you understand, Sana? Any doubt till here? Please tell me. If you have doubt, we clarify, then we'll go to the next set.
I don't think so, no.
Sorry?
No doubts.
Okay. All right. Okay. So this is alternate forms of genes. Okay. So let's see here. Okay. Alleles we have seen. Alternate forms of gene is called as allele. Okay. So now, when we talk about alleles, when we are talking about chromosomes, we are talking about homologous chromosome, homozygous, heterozygous. Okay. One second. So let's see now here. 1 minute, I'll just check. Have you done Mendel's law?
No, not yet.
Not yet, right? Okay. So what we will do is that we just go through Mendel's law step by step. We will get. Mendel was he always had interest in mathematics, biology, okay, and various subjects. Mendel is called as the father of genetics. Mendel did research on pea plant, Pisum, sativum. Pisum sativum, pea plant. Okay. After Mendel finished his experiment, he showed his experiments to the science society, but they never accepted his experiment. They would laugh at him. But after years of Mendel died, there were scientists who rediscovered his work. And then they found that what Mendel had given was like a treasure. And that's how Mendel came to be known as the father of genetics. Okay. Mendel, he took few, important characters. He took contrasting characters. One example I will give you. He actually took 7 contrasting pairs of characters. So one I will give example. He took a character which is, let's see, height of the plant. In height of the plant, there are 2 traits. See, this is character. Height of the plant is a character. And we have 2 traits. 2 traits here. Height of a character, 2 traits. One will be tall. Another one is dwarf. Okay, tall and dwarf. So tall is a trait and dwarf is also a trait. Height of the plant is the character. Okay, height of the plant is the character. Like this, he took 7 contrasting characters and then he devised the different type of cross involving single gene, mono hybrid cross involving 2 genes, called as dihybrid cross. Okay,
Yeah.
what cross? Dihybrid. Monohybrid involves single gene, dihybrid 2 gene. So let us see one monohybrid cross. Okay, let us see what monohybrid cross. So monohybrid cross, Sana, involves 1 gene or 2 gene?
1.
1 gene. So what we will do is that we will take height of the plant. We will take tall plant. This is dominant trait. Height. This is character. Tall is dominant trait. Dwarf is recessive trait. So we represent tall as capital letter T, capital A. Why 2T? Because we have alternate forms of genes. See, the chromosome I told you, right? Alleles. This capital letter T and small letter t is nothing but alleles. Alleles are alternate forms of gene. Okay, alternate forms of gene. Okay, next, let us see here. So what we will do is that we'll take the parental generation. The parental generation that he took for the plant, he always made sure that it is a pure line. Pure line meaning homozygous. Okay, homozygous. We are able to, we are able to understand this in a homozygous means, you can say like this: capital letter T, capital letter T. This is tall. Small letter T, small. This is dwarf. This is homozygous, dominant. This is homozygous dwarf. Now from this, we'll get gametes. We'll get what? Gametes. Now, gametes, capital letter T, and draw your small letter T. Then we carry out fusion of gametes. So we get capital letter T, small letter T. So this is called as the F1 generation. F1 refers to first filial generation. First filial generation is a combination of both the parents, one dominant and one recessive. So F1 generation is always, first of all, because capital letter T, dominant, and not only dominant, but it is always hybrid, mixture, capital letter T and small letter T. This is the first filial generation. This is clear, Sana?
Yeah.
Okay. Now what we do is that we want to see F2 generation. We want to see the F2 generation. So hybrid we will self. Now here we will use a method which is called as Punnett square. The Punnett square will help us to get the F2 generation. So we have right here T, T. We'll put here another capital letter T, small letter T. Okay. So can you tell me what will be here, Sana? Capital letter T, small letter, what will be here?
TT, like big TT.
Yes, one capital, one small.
Yeah.
Here also one capital, one small. And this is both are
That's small.
small.
Yeah.
So in F2 generation, we have capital letter T, capital. And if you remember, this is also present in the parents. Capital letter T, small letter T, we have two. This is the hybrid. Small letter T, small letter T, homozygous. Okay. So how many capital letter T did we get? This means it is tall. What do you mean? What do you understand by this? Capital letter T, small letter T. Is it tall or dwarf?
Tall.
Yes?
Tall.
It is tall.
Tall.
Yes. What about this capital letter T, small letter? That is also tall.
Yeah.
What about small letter T, small letter T?
Small.
Dwarf, right?
Mhm.
So, when we say phenotypic ratio, phenotypic ratio. Phenotypic means external morphology. External morphology. So how many homozygotes? Capital letter 1. This is 2. Sorry. How many are tall? 1, 2, and 3. How many is dwarf? 1. So phenotypic ratio is 3:1. So out of 4 plants, 3 plants are tall and 1 is dwarf. So we write 3:1. Next. Genotypic ratio. Genotypic ratio. Okay, genotypic ratio is the genetic makeup. Okay, genetic makeup. So Sana, can you tell me what would be the ratio?
3:1.
Yeah, sure.
3:1.
Sorry?
3:1.
3:2:1. Yes or no?
Oh yeah.
3:2:1.
Mm-hmm.
This is Mendel's monohybrid cross. If we see dihybrid cross, then it's the inheritance of 2 genes. Okay, it would be the inheritance of 2 genes. Then, okay, so this point is clear, Sana. Any doubt here?
No.
Sure?
Yeah.
Okay, genetic makeup. So, when we are talking about the genes, we know that genes carry genetic information and they are present in the DNA. Now, in our DNA, we have base pairs, or whenever the DNA is forming, mutation can occur. Sometimes when a person is exposed to too much of radiation, mutation occurs in the genes. Mutation. Mutation. Can you define, Sana, mutation?
When like there's
Yes,
like
tell me. Define in your own words whatever you know about mutation.
it's when like
Some kind of change in the nitrogen base pair.
yeah.
Yes, some kind of change in the DNA.
Mm-hmm.
Right? Mutation are changes in DNA. And these changes can be due to chemical mutagens. They can be due to physical mutagens. Chemical mutagens. Chemicals that bring about mutation. Okay? Chemicals that bring about mutation. Physical mutagens like X-rays, radiation, X-rays, gamma rays. Okay, all these. Okay, then let's see. Okay, so mutation. So mutation will be changes in the DNA. Changes in the DNA. This mutation can occur at any time spontaneously, but our respiration system has to correct it, has the ability to correct it. Some mutations can be corrected, but while others are very harmful, it may kill the person as well. Okay, is it clear?
Yeah.
Okay. All right. Now, let's see here. Okay. Let me see here. What else are there? All right. So, mutations can occur in cells as well. We have a type of cell which is called as proto-oncogene. Proto-oncogene. This gene is present in all of us. Okay, this gene is present in all of us. Proto-oncogene, due to some kind of mutation, causes or forms oncogene. Proto-oncogene is the normal gene. This normal gene undergoes mutation to form oncogene, and it is this oncogene that will cause cancer. Clear or not?
Yeah.
Yes?
Clear.
Okay. Let's see. Now, we see here about a mutation which is called as point mutation. Okay. Okay. We'll see something called as point mutation. Do you have any idea what is point mutation?
No.
Yes?
No.
No? Sure?
Yeah.
Yes? Okay, let's see. Okay, 1 second. All right. Okay. So, we'll see what is point mutation. So, point mutation is change in single base pair. Okay? Change in single base pair. Is this clear?
Yeah.
Okay. So, let's see here. Okay. All right. Let's see. Okay. Point mutation. Changes in single base pair. Okay, it's called as point mutation. And the type of point mutation that occurs in sickle cell anemia is substitution. Okay, substitution. Okay. Now, let's try to understand. Let's try to see this here. Okay, so how does sickle cell anemia occur? Okay, sickle cell anemia. Now, we have in our hemoglobin. You know what is hemoglobin, right?
Yeah.
Yeah, we have A peptide chain and we have B peptide chain. Okay, so whatever the changes or mutation that occur is in the B peptide. Okay, whatever is there in the B peptide. So what happens? The sixth amino acid, okay, sixth amino acid becomes valine instead of glutamine. Okay, It's valine instead of glutamine. Actually, glutamine should be present, but it is substituted by what? Valine. Okay. So here the sixth amino acid in the chain is valine rather than glutamic acid. Glutamine or glutamic acid. Glutamine or glutamic acid. This point is clear?
Yeah.
Okay. Now, let's see. Okay. So, hemoglobin. All right. So, only one substitution. So, since it is one substitution, this is called as point mutation. Due to this point mutation, what happens? The RBC becomes sickle shaped like this. Okay, this is the RBC acquiring a sickle shaped structure. Okay, sickle shaped structure. So as a result, what happens? Okay, sickle shaped structure. So due to which what happens? The oxygen carrying capacity of RBC decreases because RBC appears sickle shaped. So here definitely the RBCs which are sickle shaped, they cannot, they cannot carry oxygen. So what happens in such case, sickle cell anemia, even there is decrease in the amount of oxygen in the body. Okay, oxygen in the body. So it is sickle shaped. Okay, all right, sickle cell anemia. Okay, now let us see this. Okay, this is the physiology of the disease. Now we'll see this disease from the genetics point of view. Let us see this disease from the genetic point of view. Sickle cell anemia. This is autosomal recessive. Autosomal recessive. That means autosome means other chromosomes other than the sex chromosomes. Chromosome other than sex chromosome. Chromosomes other than sex chromosome. Clear?
Yeah.
Chromosomes other than sex chromosome. That is called as autosomal recessive. And recessive, recessive, when it's not able to express itself. Autosomal recessive disorder. So here, okay, here what happened? It can be HPA, okay, and HBS. HPA and HBS, okay. Now, what happens here? Let's see. HPA, okay, the disease is controlled by a single pair of allele, HBA and HBS. So here, HBA, this is normal. HBS is the gene, is the defective gene. HBS is the defective gene. Is this clear? Okay. Now, one second. Okay. So, for example, if a person is HBA and also HBS, okay, HBA and HBS. So here, how many alleles are the one that is, causing the disease? One, only one, or both?
Both.
See here, HBA normal, HBS defective gene. So, yes, tell me. These are heterozygous. These are heterozygous. Okay, these are heterozygous allele. That means it has HBA, which is normal, and HBS, which is defective.
Mm-hmm.
Okay, heterozygous, HBA and HBS. So these people are usually uninfected. Okay, these do not have the disease. Rather, they can be carriers. Carriers meaning not showing the disease but can pass to the next generation. Since it is recessive, recessive will always have carriers. This point is clear.
Yeah.
See, if you have not understood, Sana, I'll explain you again. You tell me whether it is clear or not. Tell me where you missed. See, you read. You take your own time. You read.
Yeah.
No problem. No hurry. You read. But tell me if you have understood or not. Because if you have not understood, I will explain you. You know, genetics is one subject where you don't understand even a small thing that piles up and you don't understand the remaining things.
Yeah.
Right?
Mm,
Can you just tell me?
I think I understand it, but could you explain the carrier part again? Like the
Carrier part, yes.
yeah.
But you're seeing the carrier means, since it is the disease is autosomal recessive. Recessive means what? Can it express itself? Can it express itself? No.
No.
Autosomal. What is autosome? Chromosome other than the sex chromosome. So
Yeah.
in the autosome, we have a recessive gene. This because there is one recessive gene and one normal. See, HBA, HBS. Can you see that?
Yeah.
One normal, which is normal. HBA is normal. HBS is affected. Okay. So we can call this individual as a carrier. That means he or she himself will not show the disease. You understand?
Mm-hmm.
Because they will not show the disease because they have one normal allele and the other affected allele and both are present together. So they will not show the disease. They have the defective allele, but they have only one defective allele. So they will not show the disease. Rather, they will pass on this allele to the next generation. This point is clear?
Yeah.
Now, did you understand?
Yeah.
Okay. Heterozygous. Okay. But suppose if a person has HBS, then what is this? Affected, uninfected, carrier or affected? Carrier or affected? HBS, HBS.
Is it effective?
Yes, it is affected.
Mm-hmm.
Homozygous. Because the allele, HBS is the allele for sickle cell anemia, but it is recessive. So to express the disease, it has to have both the allele which is recessive. If there is one dominant, one recessive, only one, then it's a carrier. Here both are having the affected allele. So that individual is what? Affected. Okay, this point is clear?
Yeah.
Okay. This is sickle cell anemia. Decrease in the amount of oxygen. Okay. All right. Next. Okay. When we are talking about chromosomes, We know that chromosomes are the carrier of genetic material. Okay. Chromosomes are the carrier of what material? Genetic material. Let me see this. Okay. All right. 1 second. Okay. All right. So now what we will see is that let us see chromosomes in the lower forms. So we know chromosomes are carrier of genetic information. Chromosomes carry genetic information. So we'll now study different type of chromosomes. Chromosomes in the prokaryotic cell, chromosomes in the eukaryotic cell. You know what is a prokaryotic cell?
Yeah.
Yes? Yes. Okay. Let's see this. First, we will come to the prokaryotic cell. Chromosome in prokaryotic cell. Prokaryotic cell is a bacteria. Prokaryotic cell is what? Bacteria. Now, if we draw the structure of the bacteria, okay. All right. This is called as the nucleoid or incipient nucleus. Why? Because it is none other than bacterial chromosome. This nucleoid will have genetic information. Okay, we'll have what? Genetic information. Nucleoid. What is the nature? It is double-stranded. It is coiled. Double-stranded, it is coiled, helical coiling. It is circular. Our DNA is linear, straight. This is circular DNA. The other type of chromosome that we observe, these are called as plasmids. Plasmids. Okay, so this nucleoid is, wait, 1 second. It's chromosomal DNA. Nucleoid is called as chromosomal DNA. Then plasmids are called as extra chromosomal DNA. Okay, plasmids are called as what? They're called as extra chromosomal DNA. Because you know what happens? Because of the function of the chromosomal DNA, the bacteria will divide, binary fission. But plasmid is independent. It is not dependent on the bacteria, that once bacteria will divide, then only I will divide. No. Whether bacteria divides, does not divide, plasmid will divide. Extra chromosomal. This is double-stranded. Okay, double-stranded. It is circular. Double-stranded circular plasmids, extra chromosomal DNA. Okay, then here we have few more things to learn. Okay, now if you look into the mitochondria and chloroplast, mitochondria and chloroplast, okay, mitochondria and chloroplast, these are called as semi-autonomous organelle. Semi-autonomous organelle. Is this clear?
Yeah.
Okay. Semi-autonomous organelle means both mitochondria and chloroplast have their own DNA. Okay, own DNA. Is it clear? Yes? Right, they're not dependent on nucleus. They have their own DNA.
Mm-hmm.
Clear?
Yeah.
Okay, all right. So, Sana, what we will do is that we will continue this in the next class about genomes, the different type of chromosome numbers, sex determination. Okay, all right. Genetic variations are there. We'll do that. So, shall we end the class here?
Okay.
Yes. And any doubts, Sana, you can always mail. Okay.
Yeah.
All right. Bye. Take care. Enjoy your holidays. Huh?
Thank you.
Bye.
Bye.